And finally, in the group of molecular studies, Dr. Rob Nichols is going to be looking at miniaturization of the PWS domain for a AAV-based genetic therapy. So AAV, adeno-associated virus, is a great gene therapy vector. My background is in gene therapy, so it's a really useful gene therapy vector. It tends to be very safe. It allows long-term expression. It can get up into the brain and deliver genes to the brain. The one limitation, or one of the limitations of AAV, is that it's actually it's a small virus, so you can't just sort of throw everything in the kitchen sink into it. So in order for it to be useful for PWS and for other disorders, researchers really have to do some sort of fine-tuning and engineering of the genes that are going to be inserted and then potentially delivered. So Dr. Nichols is going to develop a novel single AAV that includes many PWS genes. So a lot of the PWS genes are small, and some of them can be sort of shrunk, and then they're going to be put together into this vector to see if that might be a way to replace the gene. So all individuals with PWS, we know, have inactive genes sitting there, and one way to address that therapeutically is to deliver active genes. And so this would be looking at delivery of active genes from the PWS region. So we're excited about this study because it is a novel gene therapy approach potentially to treating PWS. As we know, the PWS region is extremely complex, and Dr. Nichols probably knows it as well as anyone in the world because he's been studying it for many years now. So sort of that tinkering to get down to the core elements and be able to put them in an AAV is particularly well suited to his group. And we think that this would be a resource that might be useful for others. You could start looking at, how this vector, if it's delivered, for example, to a mouse or a rat model, would be able to restore function and, bring those animals towards a more typical development. So there's a lot of both in cell models and in animal models, these resources might be very useful. And Dr. Nichols is looking to use them in a beta cell model, so a model of pancreatic cells, and also in a mouse model of PWS.